All right, you guys, functions and composition of blood. So there's quite a few objectives that we have here. First, we want to describe the general functions of blood. We want to name 6 characteristics that describe blood and explain the significance of each to health and homeostasis. And then list the 3 components of a centrifuge blood sample, which you guys will get to see next week. And then define hematocrit and explain how the medical definition differs from the clinical usage. And then name the 3 formed elements of blood and compare their relative abundance. All right, so starting off with our functions and general composition of blood. Blood is continuously regenerated, and remember, it is a type of connective tissue. It's a liquid connective tissue. It moves gases, nutrients, wastes, and hormones, so it's our transportation system of all these things throughout the body. So it goes through the heart and then into the arteries, which are going to transport that oxygenated blood away from the heart. And then veins are going to transport blood that is deoxygenated towards the heart, and capillaries are going to allow that exchange between blood and body tissues of those nutrients and gases. All right, so we have some blood components. We have formed elements and then plasma. Our formed elements are 3 parts. Your erythrocytes, which are the red blood cells, are going to be transporting the respiratory gases in the blood. The leukocytes, which are white blood cells, which are going to defend against pathogens. And then our platelets are going to help form clots to prevent blood loss. Then our plasma is the fluid portion of blood. That's the matrix within the connective tissue, and it's going to contain our plasma proteins and dissolve solutes. So the functions of the blood, of course, the first one we already mentioned was transportation. It's going to transport those formed elements, dissolved molecules, and ions. So it's going to carry oxygen from and carbon dioxide to the lungs, and of course those nutrients, hormones, heat, and waste products. It's amazing how much heat our blood actually does contain. Along with that, we've got our protection. Leukocytes, those white blood cells, the plasma proteins, and other molecules of the immune system are going to help protect us against pathogens. And then platelets and certain plasma proteins are going to help with protecting us from blood loss. So another piece of the function of blood is regulation of body conditions. So the first one, of course, is the body temperature. Our blood absorbs heat from body cells, especially muscle, and then the heat is released at skin blood vessels, hence why we have vasodilation and vasoconstriction, depending on if we're too hot or too cold within the dermis. And then our body pH, blood absorbs acid and base from the body cells, and so it does have a very narrow line there of pH that it needs to contain, and so it has those chemical buffers in it to go ahead and help with that, which we talked about at the beginning of the year. Then we have our fluid balance. Water is added to blood from the gastrointestinal tract. Water is lost, of course, through urine and then skin and respiration when we breathe out. And fluid is exchanged between blood and the interstitial fluid. So that's the fluid in between the cells within the tissue or in between the tissues. So blood contains proteins and ions that are going to help maintain the osmotic balance. So there's going to be things that have a relatively negative charge compared to other parts of the body that's going to help maintain the amount of water that we have within our blood. All right, some physical characteristics. Blood color depends basically on oxygenation. So if you see blood that's super bright, it's going to have a lot of oxygen, and if it's dark, it's going to be poor oxygenated. And the volume is about 5 liters in adults. Males have slightly more than females just because they're bigger than us. And then viscosity. So blood is about 4 to 5 times thicker than water. So viscosity just means how thick the fluid is. And it depends on the amount of dissolved and suspended substances relative to the amount of fluid. So viscosity is going to increase if your erythrocyte number increases. So if your red blood cell count increases. And it's also going to increase if the amount of fluid, of course, your plasma decreases. Okay, some other pieces. Our plasma concentration of solutes, that's going to be our protons and not our protons, our proteins and our ions, is going to determine the direction of your osmosis across the capillary walls. And again, that has to do with that osmotic pressure. So during dehydration, plasma is going to be hypertonic. So fluid is going to be drawn from the tissues because you're not going to have as much water within the capillaries versus within the tissues. And then, of course, our temperature. Our blood is actually about a Celsius higher than what we measure at 98.7 degrees Fahrenheit. And so warmer areas that it travels through, it's going to go ahead and warm up those different areas that it travels through. Blood pH is slightly alkaline as well, so it's about 7.35 to 7.45, and that's that really narrow range that it needs to maintain for our pH balance. And that's really important to make sure that our proteins don't become denatured, because otherwise they wouldn't work, and then we would be quite screwed. So components of blood, when we centrifuge blood out, there are three parts. We've got our whole blood, which is basically the plasma and formed elements that we can separate. So, there's three parts within the whole blood, okay? So the first one is going to be the plasma. That's that yellow color that's at the very top, and it's about 55% of a sample. And then you've got the buffy coat, which is really thin. It's about 1%, and it's a gray-white color, and it has the leukocytes and the platelets, so those white blood cells. And then the very bottom, the red one, is going to be the erythrocytes, or the red blood cells, and it's about 44% of the sample. And that's what we're going to have some fun going ahead and looking at, just to see, that our plasma is going to have mostly water within that 55% of whole blood, and then a lot of proteins, which are going to be those parts that go ahead and help with our immunity, and then of course those other pieces like electrolytes and waste products and nutrients and those kind of things. And then the buffy coat is really pretty awesome as well, because it's got some really powerful cells within it called leukocytes that are going to be helping to go ahead and defend against anything that comes into our body that's a foreign pathogen. And then platelets, of course, are blocking and helping reduce blood loss when we go ahead and have an injury. And then that last part is going to be those erythrocytes, which is pretty crazy. If you notice here, 4.2 to 6.2 million erythrocytes, little blood cells per cubic millimeter, which is a lot. The other part with centrifuge blood is talking about hematocrit. This is the percentage of volume of all formed elements. So the clinical definition is the percentage of only erythrocytes. So if someone says, here, your hematocrit is this, it's just telling you how many erythrocytes in your centrifuge blood, or the percentage of erythrocytes in your centrifuge blood. So for adult males, it's about 42 to 56%, and for females, it's 38 to 46%. So notice us ladies actually have quite a few, quite fewer erythrocytes than our male counterparts, and that's just because testosterone causes more erythropoietin secretion by kidneys, and that's just a hormone that's actually going to be stimulating erythrocyte production. So a blood smear, this is just a thin layer of blood that's placed on a microscope slide and is stained, and you can look at the different appearances of the formed elements that we find in blood. So our erythrocytes are going to be, of course, the most numerous. They're going to be pink, anucleate, so they're not going to have a nucleus, and they're going to be biconcave discs. And leukocytes are going to be much larger and varied in form, and you'll see noticeable nucleuses. And the platelets are just little fragments of cells, which is just kind of interesting. So we can see here some of the pictures that where those leukocytes are, those big purple guys, and then of course the red blood cells are the most common there, and the platelets are itty bitty little fragments there in the corner. All right, so what did we learn? What are the functions of erythrocytes and leukocytes? So erythrocytes, of course, are binding to oxygen and carrying carbon dioxide. Leukocytes are going to be protecting our body from foreign pathogens. And would a hematocrit of 39% be considered normal? For us ladies, yes, but for men, no. And then what is the pH of blood and why does it matter? It was that 7.3 to 7.4 range. And this matters so that we don't go ahead and have denaturation of our proteins that are in our plasma, which would then, of course, be bad for all the things that proteins do in our body. All right, thanks, guys.